Welcome to the PubMed Daily Review for April 22nd, 2026. Today we scanned 2,715 new open access papers. Here are the top 5 selected for detailed analysis. Trend 1 of 5, Oncology and Organ Health. This trend explores the interplay between cancer treatments and organ function, focusing on patient outcomes and therapeutic strategies. This cluster covers 529 papers. Representative work includes global research on the crosstalk between microbiota, intratumoral microorganisms, and liver cancer, a visualization analysis, echocardiography in cardio-oncology, optimizing service delivery, and pelvic lymph node dissection in prostate cancer, current evidence, controversies, and future directions. Trend 2 of 5, Social Determinants in Health. These papers explore how non-medical factors influence health outcomes and healthcare delivery. This cluster covers 536 papers. Representative work includes identification of spatiotemporal associations of social determinants of health on the incidence of adverse birth outcomes in Louisiana, reproductive health, a blind spot in psychotherapeutic treatment, evidence of insufficient consideration of reproductive factors in routine care, and the combined predictive value of the triglyceride glucose index and a body shape index on all-cause mortality. A 26-year prospective cohort analysis from the Rancho Bernardo study. Trend 3 of 5, Neuroscience and Gut-Brain Axis. These papers explore the intricate bidirectional communication between the nervous system and the gut, influencing health and disease. This cluster covers 278 papers. Representative work includes dimorphic neural network architecture prioritizes sexual-related behaviors in male Cerebditis elegans. Tannin phytocomplex improves IBSD symptoms and gut-brain markers. Prospective pilot study and correction. In vitro differentiation of the hypothalamic Kindi neuron, a master regulator for reproduction. From mouse embryonic stem cells. Trend 4 of 5, Cardiovascular and Transplant Outcomes. These papers investigate outcomes and interventions related to cardiovascular and transplant conditions, often employing advanced imaging or clinical analysis. This cluster covers 631 papers. Representative work includes determining a gamma knives isocenter using a starburst shot, a potential novel molecular interaction in bronchiolitis obliterans syndrome in lung transplantation patients, the role of serpina-3 in osteoprotegerin, and surgical aortic valve replacement in severe left ventricular dysfunction, mortality, echocardiographic, and strain outcomes. Trend 5 of 5, Molecular Mechanisms and Spectroscopy. Investigating molecular processes and their spectroscopic signatures across biological and chemical systems. This cluster covers 741 papers. Representative work includes the PAR/delta-induced mesenchymal stromal cell secretome has cytoprotective effects via ANGPTL4 in a preclinical model of acute lung inflammation, toward stimulando time-resolved infrared spectroscopy to study intermittent light-stimulated CO2 hydrogenation and structural mechanisms of pump, assembly, and drug transport in the ACRB-TOLC efflux system. Up next, our first paper out of 5, Precise Specific Gene Editing via a Compact GoCas12M, FokI Chimeric Nuclease by Tin Marsic and Magdia Mahfouz. Let's break down its key contributions. CRISPR gene editing has revolutionized our ability to modify DNA, opening doors for functional genomics and potential therapies. However, current technologies often struggle with delivery challenges and unwanted off-target edits. Many CRISPR systems are simply too large to fit into common delivery vehicles like AAVs, limiting their therapeutic reach. This study explores a novel approach to overcome these limitations. To address these challenges, researchers developed a novel gene editing platform by fusing a compact, catalytically inactive Cas12M protein with a FokI nuclease. This GoCas12M FokI system utilizes a dual-guide RNA strategy to bring 2 nuclease units together, enabling precise DNA cleavage. The study tested this system in human cell lines and on several endogenous human genes, evaluating its editing efficiency and specificity. The results were highly promising. The GoCas12M FokI editor showed robust and efficient editing at multiple human gene targets, including HBB and AIFM1. Crucially, extensive analysis confirmed no detectable off-target edits, indicating a high degree of precision. Furthermore, The engineered editor is significantly smaller than conventional systems, making it compatible with AAV vectors for improved delivery. This study presents a significant advancement in genome editing technology. The GoCas12m FokI system's compact size and remarkable specificity make it a powerful tool for therapeutic applications, particularly in gene therapy, where efficient delivery is paramount. While these findings are exciting, further research will be essential to fully explore its potential and ensure its safety and efficacy in clinical settings. Up next, our second paper out of 5. RBFOX1 dysfunction unlocks APO for associated microglial genesis and exacerbates Alzheimer's pathology in human cerebral organoids by Bowen Zhang and Ru Zhang from Institute for Regenerative Medicine, State Key Laboratory of Cardiology and Medical Innovation Center, Shanghai East Hospital, Shanghai Key Laboratory of Signaling and Disease Research, Frontier Science Center for Stem Cell Research, School of Life Sciences and Technology, Tongji University, Shanghai, China. Let's break down its key contributions. Alzheimer's disease is a devastating condition, and we know that a specific gene variant, APO4, significantly increases a person's risk. While other genes are also implicated, like RBFOX1, how they work together, especially with APO4, isn't fully understood. This study aims to shed light on this complex relationship by investigating the functional interplay between RBFOX1 and APO4. To tackle this question, the researchers developed a sophisticated model. They use human cerebral organoids, essentially mini brains grown in a lab, and engineered them using CRISPR technology. They created organoids with either APO3 or APO4, and then knocked out the RBFOX1 gene in some of these. This allowed them to directly compare the effects of RBFOX1 loss in the presence of different APO variants. They then employed a range of advanced techniques, including RNA sequencing and specialized imaging, to analyze the cellular and molecular changes. The results were quite striking. When RBFOX1 was depleted, it specifically triggered a significant increase in microglial cells, but only in the organoids carrying the APO4 gene. These newly generated microglia showed signs of being neurotoxic, secreting inflammatory factors, and impacting synaptic structure. This was accompanied by a worsening of key Alzheimer's hallmarks, like tau hyperphosphorylation and abnormal electrical activity in the organoids. Essentially, RBFOX1 appears to act as a brake on APO4, driven microglial activation and subsequent neurodegeneration. So, what does this mean for Alzheimer's research and potential treatments? These findings highlight RBFOX1 as a potential protective factor, suggesting that its loss exacerbates AD pathology, particularly in individuals with the APO4 variant. This work also introduces a novel and powerful organoid model that better mimics in vivo brain complexity, allowing for the study of gene-environment interactions. Future research could explore therapeutic strategies aimed at restoring RBFOX1 function or modulating the APO4 glia crosstalk it seems to control. Up next, our third paper out of 5. Multivariate patterns of plasma biomarkers predict region-specific Alzheimer's pathology and cognitive decline across independent cohorts by Jaffer Zamani and S. Hadi Hosseini from Computational Brain Research and Intervention, C-Brain, Lab, Department of Psychiatry and Behavioral Sciences, Stanford University School of Medicine, Stanford, California, USA. Let's break down its key contributions. Alzheimer's disease is a devastating condition marked by the buildup of specific proteins and brain cell loss. While we have ways to see these changes, like expensive brain scans, they aren't available to everyone. The big question is, can we use simple blood tests to predict these changes and future cognitive decline, making early detection more accessible? This study aimed to see if a combination of different blood biomarker measurements could predict specific changes in the brain related to Alzheimer's. They analyzed data from multiple independent groups of participants. The goal was to link these blood patterns to actual Alzheimer's pathology and how people's thinking abilities changed over time. The researchers found that by looking at patterns across multiple blood biomarkers, they could indeed predict specific changes in brain pathology. Importantly, these findings held up when tested on different, independent groups of people. This suggests that blood tests could be a powerful tool for predicting not only the disease itself, but also future cognitive decline. This study is exciting because it shows that blood tests, analyzed in a smart way, could become a key tool for predicting Alzheimer's disease. This could make early detection much more accessible than current methods. While these results are promising and validated, more work is needed to pinpoint the exact biomarkers and develop these models for everyday clinical practice. Up next, our fourth paper out of 5, Metabolic Photoimmunotherapy. A Shikonin NIR-I photosensitizer nanoplatform reprograms glycolysis to potentiate phototherapy-induced anti-tumor immunity in hepatocellular carcinoma by Weilong Yang and Yin Shun from A Department of Traditional Chinese Medicine, the third affiliated hospital, Sun Yat-sen University, Guangzhou, Guangdong Province, 510630, China. Let's break down its key contributions. Hepatocellular carcinoma, or HCC, remains a major global health challenge. Despite advances, it has a high recurrence rate, often exceeding 50 to 70%, and is notoriously difficult to treat due to a suppressive tumor environment. Current therapies struggle to overcome these limitations. This study explores a novel approach by combining metabolic reprogramming with phototherapy to potentially enhance treatment efficacy and prevent relapse. To tackle HCC, the researchers developed a sophisticated nanoplatform called PB4 at SKNPs. This system cleverly combines two agents, Shikonin, a drug that targets cancer cell metabolism, and PB4, a novel photosensitizer that responds to near-infrared light. In their lab studies, they used hepatocellular carcinoma cells and found that when these nanoparticles were activated by a 660 nanometers laser, they caused significantly more cancer cell death than any single treatment alone. This suggests a powerful synergistic effect is at play. The results were quite striking. The combination therapy using PB4 at SKNPs and laser irradiation achieved a very low IC50 of 7.5 micrograms per milliliter, indicating potent cancer cell killing. This was accompanied by significant photothermal effects, with temperatures rising dramatically under laser exposure and efficient generation of reactive oxygen species. These findings strongly support the synergistic action of the nanoplatform in inducing cancer cell death. In conclusion, this study presents a promising new strategy for treating hepatocellular carcinoma by combining metabolic reprogramming with advanced phototherapy. The developed nanoplatform shows significant potential for inducing cancer cell death and overcoming the challenges of the tumor microenvironment. While these in vitro results are encouraging, future research should focus on validating these findings in animal models to assess its effectiveness in preventing tumor recurrence and establishing lasting anti-tumor immunity. Up next, our fifth paper out of 5, Shifts in Protein Aggregate Stability Define Proteostasis Decline in the Aging Human Brain by Edward Anderton and Gordon J. Lithgow. Let's break down its key contributions. We know that as we age, our cells struggle to maintain protein balance, leading to the buildup of harmful protein clumps. This is seen in many age-related brain diseases. But what happens in a healthy aging brain? This research dives into how the stability of these protein aggregates changes over time in the human brain. To understand these changes, the researchers analyzed human brain tissue using a technique called detergent fractionation proteomics. They looked at how the insoluble parts of proteins, which can form aggregates, change with age. They found that it wasn't a simple buildup, but a complex remodeling process. Importantly, they also linked the capacity of cellular machinery like proteasomes and chaperones to the amount of protein aggregates found. The study revealed that while the most stable protein aggregates actually decreased in older age, aggregates with intermediate stability progressively accumulated. These intermediate aggregates were also found to be prone to phase separation, a process linked to disease. Even more striking, the efficiency of the cell's protein disposal and quality control systems, like the proteasome and chaperones, predicted the level of aggregate burden just as well as chronological age itself. So what does this mean? This research shows that aging brains actively remodel their protein aggregates, and the buildup of certain types of aggregates might be a critical step towards neurodegenerative diseases. This provides a new perspective on how aging impacts brain health and suggests that boosting the cell's own protein-clearing machinery could be a promising therapeutic avenue for age-related brain disorders. That's all for today's PubMed Daily. If you found this helpful, please leave a comment, and subscribe for daily paper summaries. See you tomorrow.